So good morning. Thank you for coming to the seminar. This morning we have a distinguished speaker, Brian Nault from Cornell. And Brian has been at Cornell for about 25 years. And not very often we get somebody who does a lot of applied and a little bit of basic, also some extension entomology. And we are very fortunate to have Brian here today. To just give you a little bit of background on him, he got his BS from Ohio State University, MS from the University of Georgia, PhD from North Carolina State. And again, he's been at Cornell for about 25 years. I'm going to read a little bit of information I have here. Brian and members of his program have focused on understanding the ecology of pests that attack vegetable crops, some of which transmit viruses and other pathogens. His program at Cornell has helped vegetable growers in New York and beyond, and he has brought in over $15 million to support his research and extension program. He has published about 140 peer-reviewed papers, and he has trained 14 graduate students and 12 postdoctoral researchers. Many of his former graduate students are now in academia, industry, and so forth. He has held prominent positions with ESA and has been awarded several different types of awards. I'm very happy to have Brian this morning. He's a good friend of mine and a distinguished person. Thank you.
Thank you, Ross, for a very nice introduction. And thank you so much for inviting me to come here. I've always wanted to come to Gainesville and give a seminar. And in March, coming from upstate New York, come on. I should be paying you all to come down here and do this. So anyway, thank you so much. And I had a little chance to walk around campus. And I even saw a nice big gator float around in one of your ponds. So that's pictures. At any rate, so sorry, here we go. So I'm from Cornell Agritech, and you all may go, what the heck is Cornell Agritech? So Cornell Agritech is Cornell University's largest agricultural experiment station. It's located in Geneva. And there are also 3 other smaller campuses that are now under the Cornell Agritech umbrella, spread across Western to New York to Long Island, that have faculty, but they're non-tenure faculty. Here's a nice aerial view of Geneva, New York, with it's on the the northwest side of Seneca Lake, and then the campus is right here. And we've got 4 academic units. We've got entomology, horticulture, plant pathology, and food science. And we've got maybe about 36 tenure track faculty members there. We also are home to the Cornell IPM program. We also have a couple of USDA units. And the mission at Cornell Agritech is basically to conduct research that's going to result in solutions to help growers. And our focus is basically on specialty crops, as the USDA defines specialty crops, but in particular, fruit and vegetables. I was hired 25 years ago, and at the time, there were 4 other tenure track faculty members who had commodity responsibilities in vegetables. They've all since retired. So I'm the only one. So I am the Cornell Vegetable Entomology program. And here are just some of the major foci in our program. So to study biology and ecology of insect pests, to examine the epidemiology of insects, insect-transmitted pathogens in vegetable cropping systems, to develop IPM programs, and then work with growers to implement those programs. There have been lots of approaches for illustrating the concept of IPM. I like this one because it's really simple. It gets across the point that you really need to try to use more than one tactic to manage a particular pest. But developing IPM strategies can be quite challenging. One piece of information that can influence the development of IPM programs is to have good knowledge of the agroecosystem. And what I mean by that is you need to know the crop production system. You need to know the landscape in which the pest and a crop that occurs, and also any influences of climate. Another important factor that can influence the development of IPM is knowledge of the pests and pest biology and the pathogen epidemiology. So, and of course, agroecosystem influences that. So you really need to know all of these things and how they influence IPM. And I've kind of used this conceptual framework for developing management strategies for insect pests as well as those that transmit plant pathogens. Okay, so I'm going to tell you a story today. It includes 2 bad actors and a host that'll make you cry. So you've got your onion thrips pest, which is a major pest of onion, and also a vector of a pathogen, iris yellow spot virus, that causes iris yellow spot disease in onion, also a major problem. And then the onion crop, and together, both can cause losses anywhere from $60 to $90 million per year. And if you add the cost of controlling the vector on top of that, you can add another $7.5 to $12 million per year. Big deal. Now, over the past 20 years, I've had 5 graduate students and 4 postdocs work with me on various aspects of solving this problem. 2 of them are actually University of Florida graduates. One of them sitting in the audience here, Lindsay Glacis. The other is Lydia Kamani. And then I've also worked with half a dozen faculty members at Cornell with specialties in virology and epidemiology, weed science, and then entomology. And then also working with Cornell public extension educators. Spoiler alert, I haven't solved the problem yet. But we've made some progress, and that's what I'm going to talk to you all. So here's an outline. I'm going to start off with a bit of background on the onion agroecosystem, onion thrips biology, ecology, and IYC. Then I'm going to talk about some of the knowledge gaps that we feel are needed to develop effective IPM programs, and then I'm going to share with you some of the management tactics that we've tried in New York for managing this pest. All right, so here are the areas in the United States where we have major onion production areas. You see that in general, most of the production occurs along the West Coast, in particular in the Pacific Northwest. East of the Mississippi, the 2 major areas are New York and the Vidalia onion production region in Georgia. So New York ranks about 7th or so in acreage in onion production, and about 40% of the production occurs in the southeastern part of the state, in the black dirt area, and about 60% in central and western New York. Pretty much all of the onions grown in New York are grown in muck soils. Muck soils are very high in organic matter and nutrients, and they are basically in areas where there used to be swamps. And around 1900, they were cleared and used for farmland. So onion is actually a perennial, or excuse me, not perennial, but a biennial, but it's grown as an annual crop. So seeds are taken from the scape and they're planted. The crop will grow through the season. It'll naturally mature. When that happens, the leaves fall over normally. They'll dry up, blow away, and then you're left with a bulb. That's obviously the remarkable part of the crop. Now, if bulbs remain or aren't frozen during our winters, and are we okay with that?
Yeah.
Okay.
The video wasn't showing. So
Oh, okay. I should have asked earlier. So if onions are not frozen in the winter and are very deep enough in the soil, foliage will come out in the spring, and you can have the scape and seeds and that type of thing. But it's not marketable. It's a weed essentially, and it can serve as a host for pathogens and also other insects. The onion production occurs over a 3-month span, and with planting occurring anywhere from say late March through early June, and most of the crop is seeded, with the rest of the crop transplanted by hand. And then the crop is managed really intensively with pesticides from April to August to control weeds, pests, and diseases. And then anytime from about mid-July, late July to October, you can have the crop harvest. Here's an aerial view of the elbow muck, and this is where the second largest onion production region in New York occurs. And I'm showing you this because this is where most of the onion, thrips, and IYSV research is done. If we look at the same area, but using crop data layers, the elbow muck is contained within this red area, this red circle, if you will. And you'll notice that the color here, that's onion, is pretty much the only color you're seeing within this area. And those are all onions. And if this particular crop data layer was taken, this was done in 2024. If it was done in 2004 or 1984, it looked identical. So basically, our onion growers can't produce enough onions for the demand. So they plant onions from ditch bank to ditch bank. And as a consequence, they don't rotate. So they're growing onions on top of onions for decades. So you can imagine the problems with diseases and insects. All right, onion thrips. So thrips tabaci, that is the pretty much the only onion thrips pest that we have on onions in New York. This particular insect originated from the Mediterranean. It's now common worldwide. And it does have, even though it's got a wide host range, hundreds of different species, it prefers onion. And here are the areas in red where onion thrips is the major thrips pest. So pretty much everywhere, with one exception, the Vidalia onion production region of Georgia, where tobacco thrips is the most common. And this is the type of damage they cause to the plant. So the adults and larvae will feed on the foliage. As they do so, they'll remove the chlorophyll. So the plant has a difficult time to photosynthesize, and you end up with smaller bulbs, even up to 60% smaller. And here's a perfect example of an onion field that has been ravaged by thrips. See all that foliage turning white. So onion thrips also can transmit plant pathogens. So it can transmit Irish yellow spot virus. It can transmit Pantoia annonatus that causes bacterial center rot. And then it can spread to fungal pathogens that will cause foliar diseases like purple blotch and staphylococcus wave plague. This is the life cycle of the onion thrips. It will overwinter successfully as an adult in the soil. It will emerge in the spring, lay its eggs, and most of the populations in New York are thalyticus. So this is a type of parthenogenesis where a virgin female will produce only females. And anyway, there's a couple instars before the prepupal stage, and then a prepupa goes into the soil, becomes a pupa, becomes an adult. The entire life cycle, about a month, depending on temperature. Typically, onion thrips don't begin to invade our onion fields until sometime in June, when plants have about 3 to 4 leaves. Onion thrips are very thigmotactic. That means they like the nooks and crannies. And I believe that's because they have a chance of avoiding predators and that type of thing. So depending on when the crop is harvested, you can have 1 to 3 generations completed during the course of the season. Now this is a theoretical diagram of population growth of onion thrips during the season. So as I mentioned, the females reproduce via parthenogenesis. They don't need a mate. So one female lays her eggs, and after a month, based on life table studies, about 203 female adults can result. Those 203 females produce 203 females. After another month, now we're up to 41,000 females. And one more generation, 8 million. So one female has the potential to produce over 8 million great-grandchildren. So exponential population growth, and that's going to come back. All right, we do know some potential sources of thrips in our ecosystem. After they emerge from the soil in April and May, they may colonize weeds, crank out a generation before they colonize onion. They may arrive on imported transplants from the southwestern United States. They may colonize volunteer onions in the spring. They may migrate from alfalfa and small grain into onion. We don't know the answer to that right now because the distance would have to be maybe half a mile to 5 miles to 10 miles away. But I've got a student right now using gut content analysis to maybe answer that question. So excited to find out what happens there. Okay, now moving on to IYSV. So IYSV is a single stranded negative sense RNA virus. It also has a wide host range. It causes high risk yellow spot disease and onion worldwide. And it's a tospirovirus. Close cousin is the tomato spotted wilt virus. And here are areas where IYSV has been found. Pretty much everywhere, except ironically, not Michigan or Wisconsin. I think maybe it just has a in there, but that's just my guess. And then it's not much of a problem in the Vidalia onion production region. So the virus can reduce both yield and quality. And what we find with probably most viruses that attack crops is that if the crop becomes infected early in its development, there are worse consequences on yield and harvest, right? And it can kill plants, like you can see here. It'll kill them prematurely. In this onion field here, naturally, the leaves should fall over, like I showed you in an earlier picture. Here, the plant's dying, the leaves erect. What we found is that when the plants die prematurely, they're much more vulnerable to pathogens, especially bacterial pathogens that cause bacterial bulb rot. So this field is a complete loss. IYSV transmission by thrips is very similar as tomato spotted wilt transmission. So it can be only acquired by larvae. The virus will replicate in the midgut before it then migrates into the salivary glands. It's a persistent propagative virus. It's not transovarially transmitted. The fecundity of varioleprous thrips, though, are not affected, but they do live longer, which has implications for the entomology, right? And then IYSV is not spread either by seed or mechanically. And here's just some of the temporal progression of IYSV epidemics in onion fields. This is quite a long time ago. But what you'll see is that very little IYSV occurs in June and early July, and then you can see this exponential increase. It's probably not ironic that it has that same shape as the onion thrips population growth, just saying. But what you'll see is that it can happen quite early, or you can see a delay, and then you see this. So this has some implications for management that I'll discuss in a little bit. And then in terms of potential sources, one is that it may come in on transplants from Arizona. Another could be volunteer onions in non-rotated onion fields or in cull piles. It also could occur in non-annual plant species, both biennial and perennial weeds like dandelion and burdock. And by the way, both of these are also hosts for onion thrips. And then it's possible overwintered adults. Don't know the answer to that. So that concludes the background. Now I'm going to launch into addressing the knowledge gaps that we have about onion thrips, biology, ecology, and IYSV epidemiology. So the first step I wanted to take was, well, what knowledge is important for better understanding the epidemics of insect-transmitted viruses? And I came up with these particular 5. So first is to identify the primary infection source. The second is to identify the local spread of the virus from plant to plant, whether or not or how it might be introduced in new locations, how it may survive over growing seasons, especially in New York, where nothing is happening for a large part of the season, and then factors that are influential in favoring epidemics. Well, we already have an answer to the last one. We definitely know that when there's hot weather, we have higher thrips populations, and we see a little more IYSV. We also have a pretty good idea about this fourth one. We know that we've detected the virus in volunteer onions, and also in some of the non-annual weed species. So we know that it can survive our winters in those hosts. And like I said, we don't know about overwintered thrips adults. So what I'm going to do now is talk about these other 3 areas and research that we've conducted over the years to fill in those gaps, starting with the primary infection sources. So the objective was to identify what they are in the agroecosystem, and we believe that it was imported plants, imported onion plants. And here's why. Throughout New York State, there are 2 areas where there are a lot of imported onions. It's the Elbowak area, about 35% of the acreage uses imported transplants, and in the black dirt, 15%. Where do we see symptoms of IYSV? Isn't that ironic? We don't see it in areas where they don't import plants. And so that's anecdotal information number one. The next is past studies have shown that these plants that are grown in the Southwest and imported to other locations actually may have some symptoms of the virus. Here, this Colorado study, anywhere from 0.4 to 5% of those plants had symptoms of IYSV. So the first thing that we did, this is a little over 10 years ago, is that we investigated this ourselves. So we had 5 different varieties listed there. They're shipped in tractor trailers and crates. We took the crates off the tractor trailers in March when there were no onion thrips flying around in New York. And we took those plants, we potted them up, put them in the greenhouse and thrips-proof screening, let them grow through for a few months, and then we tested them using bacillus. So overall, over 2 years we did this, we only ended up with 7 that tested positive for the virus using bacillus out of just under 1,700 plants. Pretty low, only 0.4% were infected. Okay, so maybe it isn't a major source. Well, let's do some math, shall we? So here's a 30-acre field of onions. And growers will plant typically about 130 plants per acre. So if you take the 0.4%, multiplied by the 130,000 plants per acre, that's 520 infected plants right off the get-go, times 30 acres. Uh-oh, we got a problem, folks. Over 15,000 plants starting on day 1 infected, potentially, right? So that looks pretty suspect here. And then the other item is that, and this is back in 2007, 2008, before any insecticides were applied in onions, we noticed that adults were colonizing the transplanted fields before the direct seeded ones. So there's already likely a preference, probably because those plants were larger than the seeded ones. And as I just mentioned, they're thick, more tactic, right? So you've got potential source right off the bat and a preference for colonization early on, perfect recipe for epidemic, right? So our hypothesis was that fields transplanted with these imported onions were going to be the most influential source of fostering these IYC epidemics. So I had a graduate student at the time, Ashley Leach, and she tested this hypothesis by selecting habitats where there could be potential sources of IYSV. Cull piles that had bulkier onions, ditch banks next to onions that had non-annual weed species, transplanted fields that were transplanted from onions brought in from Arizona, and then and negative control, which would be seeded onion fields. Remember, IYSV is not seed transmitted. So that would be our negative control early in the season anyway. And this is just a map of the elbow knocking areas where the different habitats were located and replicated 4 times in that 2-year study. So what Ashley did was she placed yellow sticky cards in each of the habitats and then monitored onion thrips that were captured on those cards over time, and then tested the thrips off the sticky card using RT-PCR for IYC, and it was successful. All right, and then the dependent variable here was the number of virulifrous adults estimated based on the abundance on the cards and then multiplied by the proportion that were infected with IYSV. Okay, so here are the results. On the y-axis, we have the estimated mean number of adults testing positive for the virus for the 4 different habitat types in June and July. Well, June, not much happening, but by July, well, there you go. Definitely more virulifrous onion thrips flying around those transplanted onion fields than the other habitats. So we got our answer, right? There we go. We're all set. Transplanted onion fields, we know where things are. So another graduate student came along. Well, I want to work on IYSV and thrips. Okay, Lydia. Let's actually test this hypothesis by actually sampling plants at various times in the season in seeded fields and transplanted fields. So that's what she did over 2 years. And this involved visually counting thrips on plants in the field, and then at various time points, we collected plants out of the field, took them to the laboratory, processed them, used Dacelysa to determine the percentage that were infected with the virus. And this is what she found. So starting here with thrips. So on the y-axis, we have the mean number of thrips per plant over 4 different sampling periods, which span the course of the season. But again, we're really just interested in what's happening early on, right? And here, thrips numbers, not statistically different between the seeded fields in light blue and the transplanted fields in dark blue. Huh. Well, maybe it's just a yearly thing. So in 2022, the same thing. In 2022, we had a very cool wet spring, so populations of thrips were very low. We had to add a sampling period, but the same thing. Thrips numbers early on didn't differ between the 2 plant types. But the most important thing was, well, what's happening with virus? And both years, no difference between the 2 transplant types early on. Oh, okay, well, gee, this doesn't fit with what we were thinking was going on. The other thing that was very interesting in 2021, it was a hot, dry spring. We saw thrips a lot earlier, and we saw the virus in seeded fields in late June. We never saw that earlier, or rarely, and fairly high percentage, 20%. So this suggested that maybe over winter thrips that are very leprous are actually colonizing their crop. But at any rate, so why this change? I mean, it's only like 5-year difference. Well, maybe changes in the production system occurred within that period. So I talked to the growers, and one of them, who's the largest onion grower east of Mississippi, said, oh yeah, and I'll get to that in a minute. Sorry, I'm getting ahead of myself. So typically, about 900 acres of onions were transplanted in Alabama. They started late March, ended usually maybe mid-May or so. With the seeded crop, the growers started about the end of the first week of April and finished up by the 3rd week of May. So when onion thrips were colonizing the field, say in early June, well, they were going after the biggest plants, right? Well, where were the biggest plants? They were in the transplanted fields. Transplanted fields had a huge head start on the seeded fields, right? So that made a lot of sense. Well, what happened after that is that, oh, the grower said, well, he could make a lot more money growing onions from transplants because it gives the plant a head start to produce more leaves, a bigger bulb, more money. 1,800 acres of transplants now with the same size microcrew. Well, it's going to take them a lot longer, right? So now they can still start in late March, but they're not finishing up until early June. It still can make a pretty good crop. Another thing happened with climate change. We have some springs that are actually warm, like today, believe it or not, and enough warmth that growers can actually put seeded onions in late March. They still stop 3rd week of May because you can't produce a valuable crop from seed after. But now when an onion thrips encounters an onion field, well, they can encounter a large onion plant that was seeded in late March, and they can encounter a small plant from a transplanted field that was done in early June. So now that whole seeded transplanted thing may be thrown out the window, but plant size may not. So what Lydia did was she selected a date and when she could still see some pretty significant differences in plant sizes, but at a time when we could actually pick up IYSV, so she picked a date in July, went to these fields, and we selected only direct seeded fields because we wanted to make sure we start off with no IYSV in the field, right? Gotta be fair. And she sampled plants that were large and small from fields, replicated that, used 3 different parameters: plant height, neck width, and number of leaves. And in every case, the larger plants were larger in those categories than the small ones. And then most importantly, bingo, there you go. IYSV definitely higher in the larger plants than in the smaller plants. So at 4.4-fold increase more. So even though the hypothesis wasn't true, it kind of was. We learned that basically the larger plants in the ecosystem at the time onion thrips are colonizing are going to be the ones that are going to likely be the major sources for IYSV early in the season. Okay, so we answered that question. Although we still, we're adults, get somebody soon to answer that question. Okay, now let's talk about moving into the virus from plant to plant. Okay, so the question was, can onion thrips, or do they disperse short distances from plant to plant or from field to field? Seems pretty likely that that would be true. Why wouldn't it be true, right? So that was our hypothesis that of course they'll do This is a cross section of the atmosphere. So at the top you have free atmosphere, next you have the planetary boundary layer, and then you have below the surface boundary layer, you have that particular area. So the surface boundary layer is 2.5 times the height of the surrounding topography. So any insects that are going to be flying at or below the surface boundary layer is likely to be engaging in this short distance flight. Anything above that, likely long distance flight. Okay, so had a graduate student at the time, Eric Smith, conducted this really cool study where he took these 6-meter long poles, placed them in our onion fields, and then he coated or he placed clear sticky tape. So clear because he wanted it to be passive, right? You didn't want to artificially attract thrips. You wanted to catch them just as they're naturally dispersed. So he used clear tape, and he put some at the surface boundary layer, which is 2 meters above the soil surface, and then a couple above and a couple below. And then monitored, I think, various times during the season and different times of the day, all that kind of good stuff. So just cut to the chase, this is what he found. So on the y-axis here is the altitude above the soil surface that corresponds with those areas that he had the sticky tape. And then on the x-axis here is actually the dependent variable. So the mean number of adults per sticky card on a log scale. Okay. So he found that there were significantly more thrips captured at or below that surface boundary layer than above. And in fact, 96% of the thrips were captured on those cards that were below and a little bit above. So yeah, so this is one measure. Yeah, their thrips are dispersing short distances, no big surprise, and maybe some long distance. But we wanted to follow that up with a related study and with the same question. And what we noticed anecdotally is that sometimes when you have an early maturing onion field next to one that was later maturing, that you would see damage by thrips along the edge closest to the mature field, suggesting that thrips are moving out of this because it's no longer an acceptable host, going to one that is a host. And so Eric conducted a study where he found a number of examples, like the one on your left, and then as a control, those like on your right, where he had 2 young fields next to each other. And then he recorded the number of thrips in the young field there on the left and here on the left. And he did that over 3 years. And so the data here are the number of thrips adults encountered in the young field on the left side in each case. So those next to the mature field, that would be that scenario. Here in the young field, that'd be this scenario. So again, not a big surprise. They definitely were moving short distances, and we'd see more in the scenario there on the left and the one on the right. So 54% more adults were moving to these young fields from the mature fields. And then one other piece of information is that when Eric examined whether or not these adults that were moving in the short distance were carrying the virus, he noticed a trend that the later in the season, the more likelihood you would encounter one of these adults. And that's a function of these early maturing cultivars that were harvested. The thrips, thrips move out to the adjacent fields, they get harvested. So it's kind of a concentration effect. As you go through the season, you have fewer and fewer opportunities for there to be a host, and you have more of a concentration of the virus. So that's the pattern there. So yes, I've been surprised. Thrips do engage in short distance. Okay, so we have that. And then the last one is long distance. Do they engage in long distance dispersal? And are any of these actually virulifrous? Okay, so the hypothesis was yes, they would sometimes engage in long distance movement, some would be virulifrous. And to test that, we had another project. This again was Eric Smith. We teamed up with Elson Shields, who is now retired, but at the time he was really into using these UAVs to sample pathogen spores and insects at very high altitudes. So these are remote control planes, and they've got like a wingspan of 6 to 8 feet. I mean, they're pretty big. And they were equipped with these devices here to sample biota in the upper atmosphere. So what they were basically petri dishes with a bottom cut out, thrips-proof screening put on them on the back of them. And then they were equipped with a cover here that would shield them, right, upon ascent to 50 meters above the onion field surface. And then you would want to close them on the descent, right? Because you wouldn't want to capture the threads on the way up or on the way down, because that's cheating, and that wouldn't answer your question. So there's little remote controls. So once it got up to 50, 60 meters, you clicked it, and that would open up, and you would be able to catch, and then you would close it, and then you'd land them. You did crash a number of planes. So I don't, there are a lot of flights, but it's a really cool project, and lo and behold, successful. So the total number of thrips captured, and this is 30-minute flights at a time. There are 14 different flights in 2012, 18 in 2013. So anyway, he captured thrips at those altitudes and tested some of those thrips. 20 to 15% tested positive for the virus. So this suggests that not only can they engage in long distance movement and move the virus to other many production regions, well, I mean, they can travel that and they can transmit it. So could. All right, so our hypothesis is checked out here. Okay, so all of the different knowledge areas we've at least added to or confirmed. And so now what do we do with this information? Now let's talk about management. Okay, so using this conceptual framework, knowing more about the agroecosystem and the pest biology and epidemiology, we can see if it has influenced any of the different major tactics for managing both the bacteria and virus. So I'm going to go back to this simple diagram, and I'm going to go through each of these cases, some of which were influenced by those previous studies and some not. One not is plant resistance. So colleagues of mine at USDA, Mike Havey, who's now retired, and then Chris Kramer at New Mexico State University, found the mechanisms of resistance in onion, and it involves kind of three interrelated tactics, or not tactics, characteristics. They include the color of the foliage, the waxiness, and whether or not they have high levels of low levels of, high levels of H16 ketone. So on the far left, you have susceptible onion varieties, which typically have a blue-green type colored foliage, high wax levels, and high levels of H16. In contrast, on the far right, we have resistant plants that have kind of a yellow leaf color, very glossy leaves, very low levels of wax, and low levels of H16. And then semi-glossy in between. And there are actually some commercially known varieties that fit that middle category. Several years ago, we evaluated one of these semi-glossy varieties in New York over 3 years. It was called USDA Maya, after the researcher's dog, Maya. And we compared thrips numbers with susceptible variety. So this is, I think, maybe some of the work that you did, Lindsay. And basically, most of the time we didn't see differences. Once in a while we did see a difference, but most of the time we didn't, which was very unfortunate. And the other thing that we learned with some of the semi-glossy varieties that were actually commercially available and grown primarily in the Western United States, where they didn't have the humidity and the fungal pressure we have in the East, Avalon and Delgado, we did actually see some suppression of thrips, but we encountered was far worse. Those varieties were very, very vulnerable to bacterial pathogens and fungal pathogens too, but bacterial rot in particular. The reason being, they had very low levels of wax. They're very vulnerable to the fungal pathogens. Basically melt those plants, kill them prematurely, and then the bacteria came in and took care of them. So in our climate, in the East, that particular type of resistant characteristic probably isn't going to work. Let's now move on to cultural control. Well, this would have been nice 20 years ago. Don't bring in those plants from Arizona that were infected with a virus, because that's probably how it originated, right? But it still can be a good recommendation for areas where the virus doesn't occur. Like I said, in central New York, they're not bringing in transplants from Arizona. They're growing them themselves, or they're being grown locally anyway in greenhouses. We don't have IYSV. They don't have IYSV, and they are using these greenhouse grown plants. What about harvesting early? Okay, well, if you harvest early, say in late July, using varieties like Highlander, well, you're going to harvest before IYSV is a problem. But you can't grow all of the onion acreage with an early maturing variety because it takes a lot of time to harvest these crops. And the growers on purpose stagger the maturity of the crops they grow so they can grow them over a period that can actually harvest them. So this is a good strategy, but one that is almost going to be impossible to implement everywhere, but maybe in particular areas. But it certainly can give you some ideas of where or situations where you can manage culturally. You can also use this information another way. So early on, I was asking growers where they would plant their varieties, and there was an example like this, where you had varieties that would be harvested in July in yellow, those in blue in August, and the real late ones in September in purple. But kind of wherever they wanted to grow them without any real thought. Well, we know from what I just showed you that when a crop matures, what happens right beforehand? Those strips move out to the next fields. Well, in this particular scenario, 50% of those fields that aren't being harvested are going to be vulnerable to infestation by these virulifera strips that are moving. Well, what about this? Organize them in a more strategic way so that when thrips are moving out of those mature fields, they're going to just the next one, short distance dispersal, and the other ones are going to be spared, at least for the time being. So here, this scenario, only 12.5% of the fields are going to be maybe vulnerable. Plus, you know what direction to come. Another idea is to minimize plant stress so that the symptoms or the consequence of the virus on the plant isn't as bad. And water management is huge. So not only irrigation is important when it's hot and dry, but more importantly, to be able to get water off after big rainfall events. When we get remnants of tropical storms, which we do, believe it or not, in New York, you can get these 3 to 5 inch rainstorms in July. And let me tell you, right afterwards, you could almost see the symptoms on the plants overnight. It's unbelievable. The stress that they go under, wet feet, they don't like wet feet. So being able to pump that water out of those low areas is important. First, they're grown in these, there used to be swamps, right? So it's already low to begin with. And then maybe optimizing plant health. There hasn't been a lot done in optimizing nutrient management. Maybe that could be another factor to help. Moving on to biocontrol. Well, I haven't really examined biocontrol per se, but I have investigated maybe the potential of naturally occurring biocontrol with predators that have an impact. And certainly you could find natural enemies. Here are some of the more common ones here: coccinellids, syrphids, chrysophids, antichorids, and some predaceous thrip species. But what we see is this, that the densities are positively correlated with thrips populations. So when you see real high thrips populations, which are way above threshold, well, that's when you start to see the predators. Well, at that point, they're not doing anything. So I really don't think that at least relying on naturally occurring biocontrol is going to have a practical impact in the current system. And I'm going to finish up with chemical control. So we have in place really good guidelines for managing onion thrips populations with insecticides. And they're based on insecticide resistance management principles. We have effective products belonging to different classes, like maybe 7 different classes. We have developed action thresholds for these products. Generally, products that are maybe average or fairly good are going to have pretty low thresholds. Those that are really effective, a bit higher thresholds. We have sequences of these products that can be used over time that can control thrips really well and avoid resistance development. And here's just an example of one of the more commonly used programs, where growers will start off with a systemic product like Movento. It's the active ingredient spirotetramat, usually 2 applications a week apart, and it's got really long residual activity. Usually growers can take a week, 2 weeks off, maybe even 3 before they have to resume spraying after a threshold is met. Maybe they'll use a diamide like Axerol, maybe a couple applications a week apart, and then finish up with Radiant. 6 sprays, they're done, sometimes less. If they use an action threshold, they use Axerol, let's say, and after a week, that population is below threshold, guess what? You take that one out of the system, skip a week, and then you go right into Radiant. So it works pretty well. And in fact, we followed up with a number of growers. We worked with them across New York State, and we found over several years of working with these growers that had a lot of trust in us, and that's probably the number one reason why they actually adopted this, that the number of insecticide sprays was reduced anywhere from 12 to 50%. We saved them $60 an acre, which isn't that much for a high value crop, but it's something. And no new insecticide, no resistance to some of these newer insecticides at the time the study was conducted. So pretty cool, right? So we worked with the growers, and in fact, this group received the 2019 Excellence in IPM Award, right? So we're all feeling good and dandy, right? We're doing, the extension educator was ecstatic, jumping through the fields with them. And then, so we're controlling thrips pretty well, keeping them below the economic entry level, but what in the world? We're seeing IYSD in seeded onion fields. I showed you that a little bit ago. Well, this is very unusual, given the fact that we're spraying them with insecticides. So why are we seeing onion thrips that early? And why are we seeing IYSD? That doesn't make any sense. Why is this happening? Well, maybe with climate change and mild winters, we have virulifrous onion thrips surviving the winter. They're emerging from the soil and going right into our onions. That's certainly possible, right? But still, we're spraying them with insecticides. Well, you know anything about Movento, you know that it's really good against larvae, not so much against adults. Guess what's bringing the virus to the crop? The adults. Well, basically what's happening, thank you, Brian, for your IPM strategies. I'm giving the growers a strategy that's giving half the season wide open. The virulifrous thrips come in and take care of business. So I thought, all right, let's see if we manage the crop a little differently by protecting that early period with a product that should kill adults. And maybe if we do that, we'll see on the tail end less IYSD and greater yields. So kind of a proof of concept, we had growers use Lanate, 4 applications or so, during the time when Movento was used. So we still had them use the Movento, we just had them overlay Lanate on top of it. And we did that in the Alba muck. We chose to do this in fields that were planted the same time next to each other. One field was sprayed based on the strategy that I just showed you, the standard strategy. The other field was sprayed using the intensive program, which was their standard plus the early season insecticide applications. 4 different applications. Okay, so first I'm going to show you what happened to the onion thrips populations. So over the course of the season, they weren't different in the 2 strategies. However, we're most interested in what's happening early on, right? The colonization period. Well, here you see numerically and sometimes statistically, there were fewer onion thrips adults in the intensive managed fields, which you would expect. Not a big surprise. But more importantly, what's happening with the virus. So the graph on the left are the symptoms of the virus that we saw in the 2 treatments. And on the right are the plants that tested positive for the virus using bacillus. And we saw significant reductions in both cases of the virus where we use those early season sprays. But the bottom line is that's all great, but if the yields aren't different, then it really doesn't matter, right? Well, in this particular study, we didn't see a statistically significant difference in yields. We did see a numerical increase of 7,100 pound bags per acre. Remember, I mean, the statistical power is really low, only 4 reps, right? So there's a trend here. Well, let's look a little bit more closely at each of the reps. Well, if you look at comparison of each of the reps, well, 3 out of the 4, we saw increases in yield, and only 1 we didn't. And that's why we didn't see a significant increase. So there's certainly some suggestion that maybe this is important. Reducing not only the thrips early on, but more importantly, when that virus comes in. Let's look at economics. So if you look at the cost of those additional sprays of Lanate, which is not the most expensive, it's not the least expensive product in the system. And also look at, you have to use a surfactant with all these products. And the cost of applying on a per acre basis is $15. The grower actually used 5 sprays, not 4. $200 an acre extra. Well, if you look at the average cost of a 100 pound bag of onions and the increase per acre, $1,800 an acre or more. Well, I'm not a mathematician or an economist, but I can do simple math. And that's a $1,600 per acre increase, right? In using this program. So I have shared this information with the growers. They're very excited. I saw several new trucks after I showed this. I think they're making money already and employing this program, but we definitely want to follow up and see if this is repeatable. And then finally, what about managing the virus or reducing or enhancing the plant's ability to fight off the virus so that you don't see yield loss? One of the products that has been talked about a lot is ActiGuard, and growers have used that. Researchers have used it. We just don't see any real benefit of using ActiGuard. So taking all this together, so right now we don't see any plant resistance opportunities in New York. Biocontrol, not so much, at least with relying on populations of naturally occurring predators. And I've talked about some cases where cultural control and chemical control could provide some advantages. So just to summarize, in terms of where the primary sources are, we learned that it's the fields that have the largest plants at the time thrips are coming in and colonizing the crop are going to be the more likely sources of the virus in the ecosystem. We learned that adults will disperse short distances. Number one. Number two, they're likely to move from maturing onion fields into adjacent non-maturing or younger fields. That can give growers an idea of where to look first, maybe do border sprays too in those situations, which they do on occasion. Okay. And we also know that thrips have the potential to move long distances and to colonize other areas where onions are grown. And they could potentially bring the virus to those other areas. And then finally, I just mentioned that chemical control and cultural control right now appear to be the most promising management tactics for the vector and the virus. So what's next? Well, I really want to know whether or not Euryaloptris adults can survive over winter in New York. I said that only like 1 million times during this presentation. I really need to nail that down. And also when they might emerge. That's going to be helpful for prediction purposes. The other is to identify other hosts that they may be coming in from. I mentioned that a little bit earlier too, that we have a lot of alfalfa and small grains near the onion production area. They may be moving from those areas long distance. They can do. And if that occurs, that would be valuable. Not because we're thinking that they're going to be moving IYSV, because those other hosts don't have IYSV. But if we know that a bunch of thrips are going to rain from the sky at that time, that's going to be very helpful to know. And growers may be able to focus and concentrate a little bit more during those periods. And then finally, we don't know the late period of IYSV in onion. Again, this virus is really hard to work with, and onion isn't easy, and thrips make it even more difficult. So it'd be nice to know that question, answer to that question. And then in terms of management, again, I think curtailing this early season colonization by Euryaloptris thrips is important. Using insecticides, that's just one possible tool because that's one tool that we have. But maybe in the future, some of the other technologies that have been used against thrips, RNAi, Greenlight has a product that they're looking at for thrips management, right? Perhaps a foliar type spray would be wonderful. We'll see. And also BT. There's BT being used in transgenic cotton grown for early season thrips control. Maybe if a formulation of that could be applied to onion that would work. And of course, genetically modified onion would be even better. I know both of those strategies would work a lot better, but we're talking about a vegetable crop here. Probably won't happen much. So with that, I'd like to thank all of my lab groups over the number of years. And here's just I cherry picked a couple of the years. And of course, I had to pick one. Sandy Lindsay was going to be here, so when she was in. And onion growers have really been helpful in working with us on all of these projects, a number of industries, and then funding sources from federal, state, commodity groups, industry have made this all possible. So with that, I hope there's at least some time for questions, maybe 5 minutes or so.
Yeah, maybe just questions.
I know that there are folks online as well, so I don't know how you normally do it. You allow them to ask questions first, and then you all second, or vice versa, or So, while he's doing that, you go ahead.
I think great talk. It's kind of the two parts of it really quick. So the first one is, you mentioned it's persistent propagative, but what about the inoculation time, these thrips? How is it inoculated? Because is it not on the surface of the leaf? And then the follow-up with that, do you have an anti-feed and IRAC class that can be used early season that might spread, it might reduce that initial spread?
All right, good questions. And I'm gonna try to remember both of them in order. So the first one was you talking about the persistence of what?
So you said it's persistent propagative, but
Oh,
what about
in
what
the
about-
thrips factor.
Yeah, I know that.
Yeah.
But what about the inoculation? How quick?
Oh, okay, yes,
Because from my understanding, they scrape along the surface.
That's correct.
I thought it'd be
That-
quick enough.
100%. And then after they scrape, they will then insert their proboscis in, and at that point is when the virus will move out of the salivary glands into the plant. And in terms of the time, I don't, maybe it's been investigated. I'm gonna guess like a minute or something like that. It's fairly quick. It's not as fast as a non-persistently transmitted virus like an aphid would do, where it's like within seconds, but it's probably a relatively short.
Yeah.
Yeah. So that was number one.
Yes.
And then the second one was.
With 1 minute, that's going to be pretty hard, Randy. It's whether if there are anti.
Oh, yeah, right. Yeah, but still, so out west, they use a combination of MPED, which is an insecticide soap, and Azadirect, which is azadiractive, which has anti-feed properties. They tank mix it and they apply it early in the season with the idea of being able to repel and or manage adults that would be colonized in the field. We tried that last year. The reason I didn't show you the results this year is because the grower I worked with got nervous and used some conventional products early in the season. So it ruined the experiment, but used MPED and as a directed every week throughout the season. We didn't see any reduction in trips or IFAS. But anyway, no, I still think that there is some potential usefulness in using such types of anti-feed. Yes, yeah, and we'll be looking at that one too. Brandon.
Can you separate the yield loss caused by the thrips itself from the virus?
Yes.
Because there are some strategies to have virus resistant.
Yeah, no, great question. The answer was not in this study. We couldn't disentangle them. We talked for years about maybe setting up a trial where you inoculate the plants with the virus, absent of the vector, and then really figure things out. It's a really difficult insect to work with, and we haven't done those studies. So it is true that the results that I showed, you can't necessarily disentangle yield loss caused by the thrips, yield caused by the virus. However, I didn't maybe articulate this at the time, but the thrips populations were well under the economic injury level in both of those situations. The economic injury level for the season is 2.2 thrips per leaf. They were well under that. So kind of indirectly, we believe that probably the thrips weren't causing economic loss, and it was mainly due to IYSV. I don't have 100% proof, but just, yeah. Good question.
Yeah, thank you very much. That's more than thank you.
The questions online.